So it's a pleasure for me to introduce the single but very experienced speaker in our session. It is Professor Gerrit Borchard. He will talk on enabling vaccine supply through international cooperations, lessons learned. Shortly to the person of Gerrit Borchard, he is an established academic scientist in life sciences, has a focus in his research on mucosal vaccination, protein formulation, and nanomedicines. He is experienced in both sides, in the biotech industry, where he worked as a senior executive vice president, but also as an academic and a full professor in biopharmaceutics in Geneva since 2005. He has a very high intercultural competence, including fluent language skills, has proven leadership in interdisciplinary and international team, and is a dedicated teacher and mentor. Therefore, I'm happy to give now the stage to Gerrit to give his presentation. Gerrit, the floor is yours.
Thank you very much, Stefan. Can you hear me? You can hear me?
Yes.
Okay, great. So let me share my slide here, just a second. There you go. And you should be able to see this as well, right? You see my slides? Okay, I hope you see my slides now. Thank you very much, Stefan, again, for this kind introduction. Yes, intercultural competence. I even understand Swiss German approximately 70% of the time. So following on, first of all, thanks for having me to give another presentation here at Cleanum. I had to become like 58 years old to get my own session as the only speaker in one session. But there you go. The other thing I'd like to say is that I'm not very happy to be here today because I'd rather be in Basel among friends and colleagues to give this presentation in person and not via, once again, the screen. However, I'm very thankful that we have these abilities and these options to give these presentations. And thanks very much to Beat and his team to organize this conference once again. It's very impressive. Thank you very much. What I'd like to talk about is what Patrick Hunziger mentioned this morning in terms of development of vaccines and then distributing them, because if we do not get a coverage all over the world, global coverage with the vaccinations, we will run the risk of development of new mutations, and we never get out of this pandemic. In contrast to what you heard, we are not out of this pandemic yet. So what we've done, oops, let me switch to the next slide here. So these are, this is a slide taken from Rino Rappoli's review in PNAS from 2021, where he showed the different technological advances that merged to develop these COVID-19 vaccines, which made it possible, as Stefan said, to develop these vaccines, mRNA vaccines, in a relatively short period of time. And I will come back to this. So here you see the reverse vaccinology, you see structural vaccinology, all the different developments since about 20-25 years, synthetic biology even longer than this. And we're also, as I will show you, we are also involved in a project where we try to find the ideal adjuvants for mucosal vaccination and also for certain different vaccines. And we've come a long way from the dirty little secrets that adjuvants were at the time, like in 1930, we really didn't know exactly what these vaccines were doing, to very specifically interacting with different pathogen pattern recognition receptors that we're using today. And then what Rapoli also pointed out is that not only the development of these different technologies led to the large success that we are seeing today, but also the cooperation and the sharing of data by analyzing or sequencing the genome of the virus of COVID-19 and sharing that, and then applying the synthetic gene manufacturing And from there, entering into the characterization, into the formulation, getting into the clinical trials within like 2 months' time. Viral vectors a little longer, and the proteins where you have to give also the adjuvants, it takes a little longer. But this is one of the secrets why we could so fast develop these COVID-19 vaccines, the mRNA, for instance. However, do not be fooled, please, because mRNA, the discovery of the function of mRNA is now more than 60 years old. There were 2 publications at the same time from 2 different groups that showed that in 1961. We already, and then, so we have 60 years of mRNA research, but also 60 or almost 60 years of its delivery. So the development of lipid nanoparticles or liposomes here. There was a first influenza vaccine based on mRNA formulated with liposomes already here in 1993. People keep forgetting that this has not started like in 2020, but way before that. So we are standing on the shoulders of giants, so to speak, when we're talking about the development of these vaccines. And that's sometimes forgotten. Now, coming back to the accessibility of vaccines, the global accessibility of vaccines, these are the latest data I took from this website. And these schematics are based on data from WHO as of April 27, 2022. And here you see a gross difference between the high-income countries, where we have the privilege to live in, and the low-income countries. So in the high-income countries, we have like 3 in 4 people been vaccinated at least with 1 shot of vaccination. Different vaccines, of course, whereas in the low-income countries, it's like 16.2% only. And you see this here, they're only coming up very, very slowly. Rollout in low-income countries has been very low. Low middle-income countries a little bit better. However, you see here is only like 100%, and we are here in the upper middle-income and high-income countries. We're really almost saturated with the vaccination. Now, this is, of course, as Stefan pointed out, due maybe the infrastructural problems that some of these countries do have, the distribution chains and so forth. But it's also due to the hoarding of vaccine doses by higher income countries. It's first, we were looking at ourselves to get our population vaccinated, and then we were thinking of also providing vaccines to the lower income countries. And I think this was a big mistake that has been made and is being made still. Now, what we did over only the last 5 years, no, 6 or 7 years even, is to change this a little bit by interacting, cooperating with groups in low to medium income countries and enable them to produce their own vaccines. We had a project to develop the technology to produce a pandemic H5N1 vaccine in cooperation with Surabaya in Indonesia, which was also supported by Switzerland in an R4D project. And here you see my former PhD student, Céline Lemoine, working with a team there in Surabaya. We had adjacent to that, we are currently involved in a project also financed by the SNF to test and formulate novel adjuvant and pandemic flu vaccine combinations in order to improve the efficacy of these vaccines and maybe come to dose sparing so that we do not have to apply 2 or 3 doses of the vaccine in a pandemic situation, but only maybe 2, maybe even only 1 dose to protect the population against this infection. We're currently also working on the development of polyvalent DNA vaccine against dengue fever, which is rampant in those parts of the world. It's also in cooperation with our partners at the University of Bangkok and Thailand, Chulalongkorn. And it was in 2020, in March 2020, when we in Switzerland went into lockdown in March 2020, where Chulalongkorn, our colleagues there with whom we were collaborating, approached us and said, our government asked us to develop our own DNA and mRNA vaccines. Can we do something together? And yes, of course we could, because we had a lockdown. So my PhD student and I were working in an empty building, having access to all the different equipment and different resources. And we started to formulate DNA vaccines. We got the plasmid DNA from Chulalongkorn. They shipped it over. We did the formulation, as I will show you in a minute. We did some in vitro tests. We had the physical chemical characterization. We did the tech transfer to Bangkok, where they started to do in vivo tests. In the meantime, we did some scale up. Pretty sure that we could bring this to the GMP stage. And we're currently waiting to go into the second species in order to show the efficacy. And one can dream, but go into clinical phase 1 studies as well. Now, just for your information, in case you didn't know, the central dogma of biology is you have a cookbook, which is the DNA, which contains a lot of recipes for different delicacies, these recipes are actually the RNA. And the RNA is then translated into recipes, then translated into the protein. So, and then there are not only one recipe, there's not only one cake you can make from these recipes, but a different collection of different proteins, of course. So this is the dogma. Now, there are different vaccines that you can derive from this dogma and you can use in order to transfer the information of an antigen to the vaccinee. And we went this way. We took the DNA vaccine, which then was introduced into the mice in this case, and that is then translated RNA to protein, and you get an immune response. The mRNA vaccines use the shorter way of introducing mRNA, which is then translated in proteins, and then you have the protein vaccines there as well. So what I will talk about today is the DNA vaccines that we developed here, or the formulations for the DNA vaccines that we developed here. Now, this is not something outlandish, because here you see the publication Nature in 2021, different trials on DNA vaccines that have progressed quite a bit in recent years. And this was the project that we had here, or have, is like we had the pDNA, we formulated lipoplex, so liposomes that we then complex with the pDNA, injected them intramuscularly into mice, and we looked at the spike protein expression and immune response activation of these vaccines that we developed here. We also were looking at neutralizing antibody production and also neutralization of live viruses. As controls, we were using naked DNA, so unformulated DNA, as well as electroporation as the gold standard, so to speak, for this type of DNA vaccination. Now, one of the trials I mentioned before are using this device developed by Inovio for injecting basically DNA vaccines by this electroporation method. We were using exactly this device in order to treat our mice as well, and that was our positive control, if you want. However, as we're talking about, a medium to low-income country, it was very important for our partners in Bangkok to not infringe on any pre-existing proprietary knowledge. So IP was very important to them. So we wanted to see whether we had the freedom to operate with the formulations we were having in mind. So we went to the place where Einstein was working, to the patent office in Bern. And we had a consultation with the officers there, and we made sure that we actually have FTO, so we are not infringing with our formulations any existing patents, so we don't have to pay license fees for that. And the other thing is that we wanted to, in a form of scale up, we wanted to translate the film method, that's one method of preparation of these liposomes, into microfluidics. That's the method that's been used by Moderna, by BioNTech, and so forth for making their particles as well. Want to make sure that we could scale up the whole thing, the preparation method under GMP conditions as well, make sure that this would be possible. And then tech transfer to Bangkok, in that case, from our place here to Bangkok. Now, you may be familiar with this preparation method of rehydration of a thin film layer. What you do is you dissolve your phospholipids in an organic solvent, you evaporate the solvent, you have a formation of the lipid film here, and you rehydrate with a buffer that may contain your plasmid, and you have to shake it really strongly. That was my role in the whole process because I'm a little stronger than my PhD students, so I had the task to rehydrate. Then we got a heterogeneous liposome suspension. We sonicated that, and we got a more or less homogeneous liposomal suspension. The other way of manufacturing that is microfluidics, is a continuous manufacturing method. That's why it's preferred for like large-scale production. You can make quite some batches, even with a small apparatus equipment you see here, which we borrowed actually from Precision Nanosystems while we were working on this, while we were developing this. Now we bought the system to further develop this in this project. What it does is basically you can introduce different solutions like the aqueous solutions and the phospholipid solutions in an organic solvent. You mix it in these microchannels, and then in the end you get, by self-association, you get these liposomes. So the beauty of this is that you can control a couple of these parameters here that I'm showing here. Sorry, I'm showing here. So you can control to a certain extent the physicochemical properties by optimizing different parameters, the flow rate ratio, the total flow rate, and so forth. Reproducible. It's an easy scale up. We have the medium-sized device, which we could scale up to batch sizes for clinical trials. We have, it's time saving, so we make our liposomes in like 10, 20 seconds maybe for one run. It's non-turbulent, reduction of human error because it's all automated. However, it's, especially for an academic outfit, as we are, an expensive method development. So we do a pre-formulation before we enter into the microfluidics. We cannot use all organic solvents, and there was a question about sterility, and we had to check this over and over again with every batch we produced where the sterility was still a given. So it's basically a quality by design approach when it comes to sterility. These are some of the results we got. So this is now the preformed liposomes to which we adsorb, as it is, or complex, the DNA on top of these liposomes, which were positively charged. We added the DNA, or they in Bangkok added the DNA, they mixed it quickly, and then injected them into the mice. So we did the same here. We did the physicochemical characterization. You see the Z average here and the PDI. For both the thin film hydration, rehydration, and microfluidics for different ratios, P to NP ratios between DNA and the positive charges of the liposomes between 0.25 up to 100 to 1. And you see some differences here in terms of the sizes, in terms of the PDIs, which were not so nice. So they're around 0.2, 0.4, and some even exceeding that. So we had a nice selection of different formulations which we could choose. And already here we saw a little differences between the microfluidics and the thin film rehydration. Then we looked at the zeta potential, so the charge, the surface charge of these particles, or of the complexes afterwards. And we saw here, of course, a negative charge for certain ratios, which then switched at 50 to 1, at a ratio of 50 to 1 to positive charges. For the thin film rehydration, this switch occurred for the microfluidics already at 25 to 1 ratio. So there was another difference there. We did the stability. We looked at the release of the DNA from these particles and from this assays here, and the gel retention assays. We didn't see too much of a difference between film rehydration and microfluidics. We could not identify. I know this is not a quantifiable result, but from how it looks like, it didn't seem to be much different. Then we shipped this all over to Bangkok, and they did the first studies on this. And this was the second or even the third trial we did on these mice. And here you see, this is the first dose, the second dose, the third dose. This is intramuscularly, so the naked DNA. This is the electroporation, which was the highest at the first dose in terms of total IgG, compared to the other formulations we were using. And you see here that this increase was almost the same when we were after 3 doses. We also looked at a live virus neutralization assay after we did the immunization. And here, what we saw here is actually a difference for after 2 doses between the thin film and the microfluidics. And this somewhat also was continued after 3 doses. So always the thin film formulations tended to have better results than the microfluidics results, even though we used the same ratios and so forth. Now we saw these differences, and now you get to, well, we have almost similar or comparable results. Why is it then that we see differences in immunogenicity? Now, what you have to understand is that for the characterization of these systems, we always use like orthogonal approaches. So we always use in tandem something like a microscopy method as well as another method in order to show the physicochemical parameters of this. Now, we did some TIM studies with the help of our electron microscopy department. On the left, you see the film layer rehydration, on the right, the microfluidics, and you may speculate, again, this is not quantifiable, this is subjective maybe, you might see some differences in the ruggedness of these particles. This is a little less rugged than this one here with the microfluidics. Could be an artifact, but at least we saw something. We think we saw something here and differences in between these two. And then we got together with EMPA here in Switzerland, and we used their X-ray scattering, both the wide angle and the small angle X-ray diffraction. And the spectra we got for microfluidic as well for thin film, we saw a difference here. So for the microfluidic, we saw this nice peak, actually, out of the wax. Whereas for the thin film, we always saw this little shoulder here, which was quite different from here. Now, in discussion with the guys at EMPA who ran the experiments on the wax, the conclusion from this was that if you compare the structure of the phospholipid bilayer, here A for the microfluidics and B for the thin film, we saw that the thin films, or the liposomes prepared by thin film, were a little more hydrated, contained a little more water, which also led to a higher disorder in the phospholipid bilayer, and also to a higher distance, if you want to call that, larger distance between the individual phospholipids here. It's not much here, 0.34 nanometers, and this is 0.39 nanometers. However, there are differences that we could identify by this method. Now, our speculation now, our hypothesis is that this somehow led to a different release kinetics, release pattern maybe, presentation maybe of the DNA after injection, and that in turn led to a different kinetics in terms of the immune response. And we're looking further into this. However, you see that you have to apply a lot of different techniques in order to characterize these systems, which makes them very interesting, and this work very interesting. Now, if I come now, the project we had here, or have, all the things we did. This is a table that I stole from Marcel Hoefnagel. He's an assessor for the Dutch Medicines Examination Board, and he presented this table in the CAS Strategy Forum in 2021. Now this, he talked about RNA or mRNA vaccines, so all the things that people are doing in terms of final product manufacturing characterization, release testing. So something that the authorities would expect for you to do when it comes to submitting your dossier for entering into clinical trials. Physicochemical characterization. Now you're looking at the identity of your DNA in this case, the content, the composition, strength, particle ratio, purity, impurities, potency, and safety, and so forth. And if we are looking at the project until now, we've done most of these characterizations for our formulation, for our DNA vaccine formulation. DNA particle ratio, we're almost there. We have started to do this, we're almost there. Lipid degradation, residual solvents, we have done as well. Now, almost completed that as well. And the translation in terms of a cell-based assay, we're working on that as well. So you see, also in an academic environment, in collaboration, international collaboration with a dedicated group in a low or medium income country, you can get to these results. So the conclusions we have from this, we could manufacture a stable liposomal formulation. I did not show this, but we tested the stability, so the shelf life stability in the fridge over a long period of time. That was still stable. That was also important for us during shipment, whether our liposomal formulations would arrive in Bangkok safe and sound. We looked at the pDNA complexation. They were characterized. We had different NP ratios that we selected for the in vivo experiments. We did in vitro studies looking at protein expression, the spike protein expression that was genetic information was given by this plasmid DNA in HEK 293 cells that was confirmed by confocal microscopy. We had the specific antibodies against this protein that we obtained from the platform here at the University of Geneva. So that was done as well. In vivo, we looked at neutralizing antibody production, which was observed in a certain relevant mouse model. We looked also at the inhibition of viral live virus binding. We did microfluidics, a method translation from the thin film into a method that could be used or will be used for GMP clinical trial production. And there we saw that it's not that easy to translate one of the method into the next one. You can see some surprises with this, which we also analyzed, and we got to these conclusions that I just presented. So a very interesting project. Now the key lessons, and we published that in a paper last year. Key lessons learned that are applicable to the technology transfer in terms of vaccines between Switzerland and Indonesia, Switzerland and Bangkok. What we learned is that we have to ensure feasibility, to have the need of an ecosystem to sustain the transfer technology. It's not enough to put a zetasizer in a lab in Indonesia, but you also have to train the people in order to sustain that, to work with this equipment that you put down there. The planning, local scientific infrastructure. We were very lucky to work with excellent scientists on their side in Indonesia as well as in Bangkok, and establish reliable import-export routes, especially now during the pandemic. That was a major pain in the neck to get our material into the country. Review, evaluate scientific results. So we had every 4 weeks, or we had our meetings where we looked at the different results. We needed an infrastructure to make decisions, to take decisions and work on with them. Communications is really, really important, by email, by face-to-face meetings through Zoom and so forth. Technology, as I mentioned, repetitive training on site, in location, in Bangkok, in Indonesia. So you have to train them, you have to sustain their knowledge over a certain period of time. So they're self-sufficient and can work with the equipment and with the technology you are providing, you have developed. Select materials are locally accessible, the recipient side, because we all know the disruption of the supply chains, which is maybe even worse in other countries. So you have to look into this, and you have to also look at, by the way, at cultural differences. So we had this question in Indonesia, whether we could use ethanol in order to formulate our vaccines. Is this halal or not? So we had to really also take into consideration these aspects. And to cultivate good working relationships with any funding body who would support this and support both on the Swiss side as well as on the third country side in order to overcome difficulties that are sometimes of administrative nature and less of technological nature. So now with this, I'd like to thank all these people that you see here, my team in Geneva, the antibody and imaging platforms that open up their facilities during lockdown only for us in order to help us. EMPA, Jonathan Alvaro, Leonard Kupnik, and Antonia Niels, who did the wax studies and the sex studies. On the Bangkok side, Kanita Chutito Neakachai, you see here down there. And also a big thanks to Martin Rabel, Jürgen Schmidt-Züchtig, who supported us during the lockdown by providing us a machine that we could use in order to do the first experiments there. It was a great interaction with them. One last thing, in my own interest, the Pharmacopoeia, the EDQM is organizing a symposium on the 7th to 8th of June in Strasbourg on the quality requirements for nanomedicine, which role for the European Pharmacopoeia. This is a symposium that has been inspired by mRNA vaccines. We will have the head of CMC of Moderna there. We have somebody from BioNTech who knows something about CMC. We have two people from AstraZeneca there. We have also other speakers there. Ton Lammers will give a talk. So it will be very, very interesting to look at the side of the Pharmacopoeia, how the Pharmacopoeia can help to assure the quality of these nanomedicines, not only the mRNA, but it's inspired by mRNA vaccines. Last but not least, here you have my coordinates, and I finish with 2 seconds left. Thank you.
Thank you very much, Gerrit, for this very nice presentation. Looking into the chat of questions, I think you have not only your own session with the only speaker, we are as well among us in having questions, but I will have a few of them, no doubt about. First of all, I think it was great to show in details experience where are the pitfalls and a lot of things you have to control and have a look at it if you are able to come to a final product which should be used or can be used as a vaccine. And if we look a little bit on the international market, when these new vaccines against COVID were produced, it looks like more a battlefield than another area. So many companies were not able to get through or had probably sometimes in the very end a wrong decision taken, as for example CureVac, when they just went into the final clinical data, not adapting the right dosing. So that is really one of the most important issues. So coming out from the learning and having some experience as well from our Swiss Federal Office of Public Health, as you told, it should be really organized in an international level with a lot of partners to achieve equity on vaccine deliberations. What is in your experience and with this experience with your colleagues in Asia, one of the most important advances you could take that you had really a good program which was ongoing, and which is probably not the case if I look, for example, into the Gavi aspect. They intended to send out other vaccines to other countries. Even in Switzerland, we tried to do with them, but we were rather unsuccessful, as there are a lot of limitations as well from the manufacturers in handing over such vaccines.
So, okay, a couple of questions you
Yes.
ask, as always, and you almost gave your own answers. No, but the first thing I'd like to mention is that when it comes to the introduction of the different vaccines over time, I asked the same question, or related question, to Rogerio Gaspar, who is here working at WHO. The first vaccines, we never thought, we would never dream that the mRNA vaccines would be 95, 96% efficacious. So whatever came out was approved. So they set like the threshold at maybe 50 or 60%, and we would have been happy to have something like that. Now, this is why some of the vaccines that have a lower efficacy compared to the mRNA vaccines, they were approved as well by, or recognized by WHO. Now, you cannot change this afterwards. So, we have a broad range of different vaccines, thankfully, at our disposal, with the mRNA vaccines being probably the most effective ones for now. But we do not have to forget that that might change as well. Now, what are the things to be done? Well, that's one easy answer is that what Switzerland does, and does in abundance, is to provide funding for these international corporations. When we had one of the meetings there in Indonesia, where we kicked off our interaction, there was the Swiss ambassador came. And gave a very high importance to this collaboration. So this is one of the things that, you can accompany from Switzerland, from the authorities as well, give money and support this also from a political standpoint. Now, what is also important is you have to know the people you're working with. You cannot go into a collaboration. It's more difficult to get into a cooperation when you don't know your people. You have to see them face to face. You have to eat with them and drink your coffee with them, and then you have to know that you can work with the guys. And this is what was very beneficial for us. So we both went to Indonesia, they came to us, we went to Bangkok and so forth. So this is very important. And I have to say, we were very lucky, as I mentioned as well. Bangkok, they are excellent scientists. They're really world-class scientists who are really dedicated, who are experts in their field. And it's a beautiful interaction because we have different expertises, if you want to say it that way. They have a long expertise in the vaccines. We have a long expertise in the nano characterization. So basically, that was the perfect storm until now.
Thank you very much. I think a lot of points to be addressed in that aspect, and shows as well how difficult it is probably just to start such an activity. And as you have shown in the beginning as well, how important it is that we have research and science in area where we do not see in the point when they were detected the impact on healthcare, as we have seen now with this nano and biotech issues for the vaccines, which came together in the right moment. So no doubt about that.
Yeah, but just to emphasize this, I don't know whether you have seen the newest data of BioNTech with our CAR-T trials, phase 1-2 clinical trials, where they use the mRNA vaccine to program CAR-T cells, and they had quite impressive data. So we're only at the beginning. So the mRNA vaccines, yes, nice, but that's only the beginning. I think we are at this was a breakthrough, and we are at a paradigm change when it comes to that. There will be many more of these therapies will be based on the mRNA technology and on the liposomal and nanotechnology, of course.
Yeah, just in keeping, for example, in the field of antimicrobials, having a look into antimicrobial resistance. There are a lot of very nice sessions in this program in Cleanam as well. And we know from other areas how important that is. I come back to the equity question again. Yeah. One of the lessons learned in my eyes was as well that we were confronted with new challenges regarding the logistics, especially regarding the mechanic, let's say, effectiveness or not effectiveness of these drugs. What would you, as an expert in this field, think could be one of the testings in the future to have a more reliable and quick answer if vaccine still could be used or not. We are actually at the aspect that we have quite massive amounts of stored vaccines which come to a first end of their expiry date. But it's an extreme problem to know if these vaccines can be used further or not. What is, from your experience, something which could be probably developed in that area?
Well, that's a good question because, you know, immunogenicity is hard to predict only from in vitro studies or from physical-chemical characterization or from a standpoint of even animal studies. You always get a certain indication, but in the end, you know, it's what counts is in the patient or in the persons to be vaccinated. Now we have now some some experience with that. We know to characterize the physical chemical characterization. We know what are the properties of these systems, how they should look like in order to come up with this this result. So in that point, I would rely on the data we have, the real world data, if you want, and putting them together with the information on the characterization of these systems as we have them from the PCC, from in vitro, as well as from in vivo. Having said that, we may not be able to not develop new, when it comes to mRNA vaccines, to develop new mRNAs that we have to put into this lipid particles. Now, that might be only a small change in the sequence of the nucleic acids, but this may give you, as I try to show, a difference in the physical-chemical parameters, which may then give you different results in the immunogenicity. Maybe I'm exaggerating a little bit, but to my knowledge, the regulatory authorities, they don't see that problem so much because it's just a change in the nucleic acid sequence. However, this needs to be checked and this needs to be shown if we develop, for instance, against a new mutant, Omicron mutant, a new vaccine, an mRNA vaccine with a different sequence, nucleic acid sequence, then we probably have to look again whether there might be some differences on the different levels.
Mm-hmm. Thanks. I come back even to another question. You nicely showed that we have a whole series of very different vaccines which can be used against these viral infections. Among others, there is one which is almost now authorized in Europe as well. It is the protein-based vaccine. Do you think that that kind of vaccines could be an easier way to go for low and middle income countries because of the whole infrastructure you need? Or is there again nanotechnology inside which handicaps again the whole logistics?
So the problem of why we choose for the DNA vaccines was clear, because we don't need these very low temperatures in order to stabilize the vaccines during transport. So mRNA vaccines, we tried as well, and they just degraded during transport. We couldn't do it, we couldn't stabilize it. So now for the proteins, maybe you have better possibilities to stabilize these vaccines. However, you have to produce the proteins. So what that needs is an upstream, downstream processing in a facility where you can express these proteins, purify these proteins, formulate these proteins, stabilize these proteins, characterize them. So is it easier if it's established? Okay. If it's not established, if you start from scratch now, I think my choice would more go to either mRNA vaccines, even though you have the problems with the stabilization, or to viral vaccines maybe. But proteins, they took, from my experience, I worked with proteins, we developed proteins and all the process it takes to produce them, characterize them, stabilize them. To start with this might be a little difficult. If you have some production plans for therapeutic proteins in place, that might be the easier way to go. Yes.
Okay. Thanks. With that, I think we are at the end of our session. Thanks again for this very nice presentation, opening that many questions, and hopefully there will be a very interesting meeting with the Pharmacopoeia Europe, the European Pharmacopoeia, because there are a lot of questions. I'm interested to know that as well, and hope to see you in person soon. Thanks a lot as well to the people who were there, and enjoy the cleanup meeting further on. Bye-bye.
Thank you very much.